Hello to everyone, and welcome to this talk entitled Fermentation of colostrum with probiotic microbes and phages leads to the production of GC protein-derived macrophage activating factor, GcMAF, immune system modulation, and decrease of serum nagalase activity. Before we start, I'd like to acknowledge the help of Dr. Jerry Blighty and the organizers for this great opportunity. I wish to thank also all those who will have the patience to listen to this presentation. Of course, I am obliged to advise that no information in this talk is presented by myself as a medical advice. And of course, I recommend that the standard of care for any disease must be followed, as well as rules and regulations established by health authorities of each country. I also would like to disclose my conflict of interest because, as you may know, I am the inventor of a number of supplements, and I developed the approaches and the products that will be described in this study. Here I describe how consumption of a product of fermentation of bovine colostrum, characterized by a unique microbial composition which comprises probiotics, phages, and plasmids, is associated with detoxification of metal and non-metal toxicants, decrease of serum alpha and acetylgalactosaminidase, or nagalase for short, the activity of this enzyme, decrease of serum C-reactive protein, decrease of markers specific for multiple myeloma, and a dramatic decrease of viral load in hepatitis B infection. Consumption of this product was also associated with increased excretion, that is, elimination of lipophilic and non-lipophilic toxicants such as glyphosate. Therefore, it is plausible that the detoxifying effects are of a general type. So there is a general type of detoxifying effects, and these effects are not limited to a particular class of toxicants. And finally, toward the end of this presentation, I shall propose to incorporate the concepts of brain microbiota and microbial consciousness in the ORC, or Orchestrated Objective Reduction, theory of human consciousness, with the goal of increasing its explanatory and predictive powers. So let's start with the description of this product of fermentation of bovine colostrum that is characterized by a unique microbial composition comprising probiotics, phages, and plasmids. We used a unique microbiome array, that is a very modern technique, to detect up to 12,000 different microbial strains with an extremely high specificity and sensitivity. We found that in our probiotic products, there are more than 300 different microbes, or to be more precise, DNA signatures. And this is the highest number ever recorded in a probiotic. These results go well above and beyond our most optimistic expectations. Using the tools of classic microbiology in the past, we had known for years that we had scores of different microbes. But this microbiome array, which is based on DNA techniques, revealed a richness in biodiversity that we could not even anticipate. And this is due to the fact that the tools of classic microbiology, that are the assays commonly used to assess the composition of a product as described in its label, are unable to distinguish the substrains, the plasmids, and the phages that only the microbiome array can demonstrate. These results put our probiotic approach in a league of its own, since no other probiotic can honestly make such a claim. This is because our probiotic, at variance with all other products, was designed since its inception more than 10 years ago as a means to reproduce the complexity of the healthy human microbiota. However, it is not the sheer number of different microbes, that are more than 300, that is utterly impressive, but rather the biodiversity of its proprietary formula that makes our probiotic even more unique. The genetic analysis revealed that in addition to hundreds of different probiotic microbes, it contains also a great number of plasmids and phages. And now I shall be talking about phages, or bacteriophages. Now, this is the scientific article that has been published where we describe the phage composition of this fermented milk and colostrum product assessed by microbiome array. And we also elaborate on the putative role of open reading frames that are the signaling parts of these phages in reference to cell signaling and neurological development. This article was published in the Journal of Neurology and Stroke in 2020. Now, if you go and download this article that is downloadable for free, as most of our recent articles, you find a long list of bacteriophages or phages and all their features. And we also describe how do they work. But first of all, what are phages? Phages are microbes that colonize the microbes of our microbiota. They can also be defined as the microbiota of the microbiota. And the study of phages and how they relate to human health represents one of the frontiers of molecular medicine, because phages are by far the most abundant organisms in the biosphere. Phages impact immunity directly in ways that are typically anti-inflammatory. And phages modulate innate immunity via phagocytosis, that is the ability of cells of the immune system to eat pathogens and other harmful substances, and also through cytokine responses. And they also impact adaptive immunity through the effects on antibody production and effector polarization. Phages impact immunity directly in ways that are typically anti-inflammatory. That's very important because low-grade inflammation, it is at the basis of a number of chronic conditions. And phages, therefore, they have profound effects on fighting silent chronic infections by modulating the immune response. Thanks to the presence of phages, we can now explain the excellent results that we have observed for more than 10 years with our probiotic approach in a number of conditions, ranging from autism to cancer. Now, in 2019, a number of scientific articles that you can retrieve in PubMed, which is the database of the United States Library of Medicine, they refer to phages like the dawn of phage therapy. But as a matter of fact, phage therapy is rather old. As this article published also in 2019 states, bacteriophage preparations that have a 100-year history have gone a long way in development. So phage therapy was discovered more than 100 years ago, and it is being rediscovered today using the tools of modern molecular biology. Now, in the article that I just mentioned that we published in the Journal of Neurology and Stroke, we described the effects of each strain of each phage on human health. For example, the Lactococcus phage UL36 encodes a protein that is called SAC, that is homologous to a very important human protein, that is a human recombination protein called RAD52, that plays a crucial role in DNA repair, genome stability, and prevention of cancer. And you can read the features of all the phages that we have found in our products in this article published in the Journal of Neurology and Stroke. And what about plasmids, the other self-replicating small DNAs that carry genes that benefit the survival of the organism and can be transferred to other living beings? Our probiotic not only contains a very high number of live probiotic microbes and phages, it also contains plasmids. And thanks to the presence of plasmids, the microbes are also able to communicate with each other and with the cells of the human body with mutual reciprocal benefits. And I can tell you that no other probiotic can make this claim. The composition in plasmids has been published in another scientific article in the journal Medridge Journal of Immunology. And also this article can be downloaded for free. It is entitled Natural Plasmids in a Swiss Fermented Milk and Colostrum Product, assessed again by the microbiome array. And this was published in 2019. You can download, you can see the long list of phages, the long list of those 300 and more probiotics that are present in our product. And you can also read each plasmid, what does it do, like the Lactobacillus casei strain Zank plasmid PLCA36 and all the others, very complex and rather boring names, but very, very important function for cell communication to transfer the benefits of the healthy microbes to our human cells. Now, let's move on with the other topic of this talk, how this product is associated with detoxification of metal and non-metal toxicants, often referred to as toxins, but it is more correct to call them toxicants, and decrease of nagalase activity, decrease of serum C-reactive protein, and also increased elimination of lipophilic and non-lipophilic toxicants such as glyphosate. Now, these results have been published in a number of scientific articles. This one was co-authored by Dr. Jerry Blythe, and this was published in 2017, 4 years ago. And it is a case report where we describe how intermittent fasting and probiotic yogurt consumption, of course, referring to our product, are associated with reduction of serum alpha and acetylgalactosaminidase, or nagalase in short, and increased urinary elimination of lipophilic toxicants. And this is the list of the toxicants that were eliminated thanks to the consumption of this probiotic, a long list. You can find a very long, complex chemical names. Maybe a short one is perchlorate. And all these compounds, they have noxious effects on the human body. They cause cancer, they cause endocrine disruption, they cause impairment or disruption of the function of the immune system, and so on. Also, this article, of course, can be downloaded for free, so you can go and read all the details of each one of these toxicants that has been eliminated thanks to the consumption of our probiotic. This is another article where we describe another case, another person, and in this study, we describe the changes associated with the consumption of this extremely biodiverse probiotic yogurt in a 55-year-old female from South Vietnam. In August 2019, this lady voluntarily embarked in a 3-month nutritional experience and decided to share her results with the goal of advancing scientific knowledge in the field of nutritional health. And consumption of this biodiverse probiotic yogurt was associated with a decrease in serum nagalase activity, increased elimination of toxic metals and non-metal toxicants, and a trend toward the normalization of lipid profile, like cholesterol, triglycerides, and a trend toward the rebalance of the gut microbiota. So all very, very good results. And also, this article can be downloaded. It is published in the prestigious American Journal of Immunology. It was published about 1 year ago in 2020. And here you can see from this article how the serum nagalase activity dramatically decreased after 3 months of consumption of this product. You may wonder, what is nagalase? Well, nagalase is a marker for a number of diseases ranging from cancer, as you can see from this article published by researchers in the National Library of Medicine. You can retrieve it in the National Library of Medicine through the database PubMed. And actually the title here is rather provocative. Is nagalase the key to curing cancer? And it's very interesting. Also, it's a marker for autism, as this article published almost 10 years ago in 2012 by the late Dr. Bradstreet and Emer Fogler and Linda Thayer, how they demonstrated that nagalase is associated with severity of symptoms in children with autism, and how treatment with GcMAF, macrophage activating factor, can help with those symptoms. Now, going back to our article, here you can see the elimination of metal toxicants, specifically referring to cadmium, lead, and barium, that are very toxic metals. And also here you can see the formidable decrease of glyphosate. This is so-called, it's a herbicide, and unfortunately it's widespread, and exposure of to glyphosate has been associated with a number of conditions ranging from cancer to autism. Whatever the case, the consumption of this product is associated with a dramatic decrease of glyphosate. So I think these are very good results. Now, I was also mentioning C-reactive protein. C-reactive protein is a very old and rather non-specific marker of inflammation, in particular low-grade chronic inflammation. In this other article published in the American Journal of Immunology, we observed again decrease of nagalase and also decrease of C-reactive protein values in another subject. This article was co-authored by Dr. Michael Carter and Dr. Stefania Pacini. In this article, we also demonstrate how this probiotic product of ours contains about 100-fold more GcMAF activity than purified chemical GcMAF. Again, it's a very complex article. You can go and read it in detail, but the main message is that this probiotic contains 100-fold more GcMAF activity against human nagalase in comparison with purified chemical GcMAF. And by the way, our product is 100% natural. Now, what about multiple myeloma and the viral load in hepatitis B that I've mentioned before? All these results have been published in scientific articles, peer-reviewed. Now, this has been published again in the American Journal of Immunology in 2019, together with Dr. Nicola Antonucci, who is an expert in autism research, but in this case, it refers to a lady with multiple myeloma. And in this case, the use of an extremely biodiverse probiotic is associated with significant decrease of markers for multiple myeloma, which is a serum-free kappa light chains, as well as a trend toward the normalization of the kappa lambda ratio. Again, these are specific markers for this type of a cancer. And also decrease of plasma cell bone marrow infiltration, in this case of multiple myeloma. And again, you can go and read all the details of this clinical case in this scientific article. As far as the dramatic decrease of the viral load in hepatitis B, here you can see how in about 3 months, 3 independent laboratories demonstrated how in a gentleman, the viral load went from millions of copies down to 10,000 or less. So this is an extremely impressive decrease in a viral load in a case of hepatitis B. Also, this has been published. You can go and check this document object identification. So this DOI, and you can read all the details of this case. And we are towards the end of this presentation. And here I propose to incorporate the concepts of brain microbiota. We have microbes in our brains. We have microbes in our brains just as we have microbes in our gut, in our skin, in our mucosa, essentially everywhere in our bodies. So we also have microbes in our brains, and those microbes in our brains, they exert a number of very important functions. And I propose to incorporate the concept of this brain microbiota of ours and microbial consciousness, microbes, they have consciousness, into this theory of human consciousness that is called ORC. ORC stands for orchestrated, OR stands for objective reduction. And the goal of my proposal is to increase the explanatory and predictive powers of this theory. Now, this goes back to a few years ago, when in 2017, I published this article in the Madrid Journal of Immunology entitled Human Microbiota and the Immune System, Reflections on Immortality. So it's an article that is half pure molecular biology and biochemistry and microbiology, and the other half is rather philosophical or speculative. But from speculation, now we go to real hard science, talking about this theory of orchestrating objective reduction. I wish to state that the founders of this theory are Professor Stu Ameroff from the Arizona State University in Tucson, Arizona, and Sir Roger Penrose, who just recently has been awarded the Nobel Prize. So this theory has a Nobel laureate as a founder. Now, the orchestrated objective reduction theory attributes consciousness not to synapses and to the web of connections inside our brains, as it is commonly understood, but to quantum computations inside the microtubules that are subcellular structures, like small tubes inside our cells, inside brain neurons. And you can go to Wikipedia or you can go to PubMed and read tens, if not hundreds of articles concerning this extraordinary theory that explains the mystery of consciousness. I published this article just a few days ago, and this article is now in press in the Journal of Neurology and Stroke, because of course it refers to neurological issues, and it is entitled "The Brain Microbiota as it relates to the OR theory of consciousness." Now, in this article, I write that the OR might indeed be the most complete theory of consciousness, even so it has to be considered special. Special means like a special relativity, limited, because it is relative to the human part of the brain. Now, if the concept of the brain microbiota, that is the microbes in our brains, and the deriving concept of the rhizome, as it has been described in the article that I've mentioned before, if those concepts could be integrated in a more general OR theory, its explanatory and predictive powers would be vastly increased. For example, the OR theory postulates that consciousness may exist independently of biological substrates. So we may have consciousness independently of neurons, without brain. Can we have consciousness? Can we function without brain inside our skulls? The answer is yes, because there are cases of people with vastly reduced, almost with no brains, who lead normal lives. And this appears to support this postulate that consciousness actually is not inside the brain, it is inside the microtubules. Now, this is a rather provocative statement that we can live and we can function very well without brains, but this has been very well demonstrated many, many years ago. This article was published in the Lancet, arguably one of the most prestigious medical scientific journals. And here, researchers from France, they demonstrate with the MRI, magnetic resonance imaging, the almost complete absence of a brain in a white-collar worker, a person who was functioning extremely well in the French city of Marseille, driving his car, having his family, working as an accountant. So a person very well integrated in society, working a difficult job without brain. And just discovering just serendipitously that he had no brain, even though he led a completely normal life. So this supports the OR theory that consciousness does not reside inside our brains, but in quantum computation inside our cells. And if the concept of the microbiota then is incorporated, so in addition to what happens inside our neurons, we also take into consideration what happens inside the microbial cells in our brains, then the issue of permanence of consciousness independently of neurons will be further supported by the consideration that the microbiota survives the human body. When we die, the microbes still live. And if consciousness is associated also with microbes, then our consciousness will still live even after our death. And this has been widely explored, this concept of a survival of consciousness, by Sir Roger Penrose and Professor Stu Ameroff. Now, if the OR phenomena occurring inside the microtubules of human neurons are entangled and shared with the microbiota, considering that the microbiota is an organ that has 10 times more cells and 100 times more DNA-based information than its human counterparts in our bodies, then a survival of consciousness, instead of being a philosophical issue, issue becomes a microbiological, an issue of microbiology, an issue of molecular biology. So I think this is very fascinating and opens a new perspective in the study of the microbes, often referred to as bugs, in our bodies, in our brains. I thank you so much for your attention. I hope you have enjoyed this presentation of mine. And again, I invite you to go and download all the scientific articles that I've mentioned, where these concepts that I've briefly mentioned are explored and elaborated in detail. Thank you so much.